Hi, my name is Joyeeta Guha, and today I'll be presenting a review on proteomic study of human cord blood reticulocyte-derived exosomes. So let's get started. So firstly, I would say that this scientific report was published in the British Journal of Hematology. Total 9 talented individuals participated in this research with Miriam Diaz-Varela and Hernando A. del Portillo. These two personalities' contribution is main since they had the hand in drafting this manuscript, analyzing the data, and even suggesting experiments to their team. So, before getting deeper into this literature, we need to understand the background of this literature. This literature mainly focuses on the protein analysis of exosomes. So, first, we need to understand what are exosomes. Exosomes are small extracellular vesicles which originate from endocytic pathways. These exosomes participate in intercellular communication, and they also have a crucial role in cargo disposal mechanism. For the introduction, excuse me, these exosomes were derived from reticulocytes. Now, what are reticulocytes? Reticulocytes are basically immature red blood cells. Okay, so and what is special about these exosomes and reticulocytes? That these reticulocytes were derived from the human umbilical cord. Now, one might ask, why the human umbilical cord? For that, I would say that umbilical cord would be the best choice because it will be the region where reticulocytes could be found in abundance. Okay. So reticulocytes are found in abundance in the umbilical cord. That's why they have chosen it. And reticulocytes are immature red blood cells, which later on mature themselves into erythrocytes or the biconcave shaped RBCs. Okay, so this study mainly focuses on the protein content of the reticulocyte derived exosomes, or in short, as we have mentioned, REX. Okay, so that it will be easier for us beginners to understand the biogenesis, secretion, and uptake of these exosomes in various biological processes. So our review mainly focuses on these questions, and as we go further into the presentation, as I go on explaining the answers to these questions will become more and more clear and vivid to us. So let's continue. So first, to understand this, we need to understand the research methods and the materials used. So isolation and culturing of the human reticulocyte-derived exosomes. This HUREX is the short form of human reticulocyte-derived exosomes. So of course, we need to isolate the reticulocytes, and isolation can be done with various processes of filtration and centrifugation. To name some of them, it is size exclusion chromatography or ultracentrifugation. For culturing, they have cultured the reticulocytes for 36 hours in presence of 1 to 3% hemocrit along with further chemicals. Nanoparticle tracking analysis. This method is used to determine the concentration and size of the exosomes. Okay, and bead-based flow cytometry. This is a very crucial process which is being used. And through this process, the scientists have identified a very special protein which is present in the exosomes. Okay, and that protein, later on, we will come to the name of it. For now, that protein is a very crucial biomarker. So through this bead-based flow cytometry, they have identified that. Immunoblot analysis and mass spectrometry, both of these methods are used for the characterization and isolation of the proteins present in the exosomes. So basically the NTA or the nanoparticle tracking analysis is used for the determination of the size and concentration of exosomes. Various immunoblot analysis and mass spectrometry is used for the same, but the proteins present in the exosomes. Here it is, one picture is given, you can see. This is basically an immunoblot analysis where they have identified TFR protein. Okay, I just named the protein. The protein is TFR, which is identified by the bead-based flow cytometry. So this protein is identified in the bead-based flow cytometry. Okay, and this is also immunoblot analysis done. And this is immunoblot analysis done, which has identified proteins present in the exosomes. And this is not immunoblot analysis. This is bead-based flow cytometry, and this is immunoblot analysis. Okay. Here they have identified TFR, which is a biomarker. And here they have identified the proteins present inside the exosomes by immunoblot analysis. And this is by bead-based flow cytometry. Okay. So the new techniques in instrumentation which is used, mass spectrometry based proteomics. So in proteomics, various processes have been used earlier, but this was the first experiment where mass spectrometry was incorporated. Size exclusion chromatography, in short, we all know this as SEC. This is used to obtain the sample in its purest form, and it is a very useful method. Immunoelectron microscopy, this is used to produce very high resolution images to determine the diameters, the sizes, and the distribution of the exosomes in the sample. Functional assays include the ultracentrifugation and the processes which are required for the isolation of Eurex. Okay, those are the functional assays. And through gene ontology analysis, we have identified the proteins which are present in the exosomes and which play a crucial role in the living body. So now we come to the most important section of the presentation, which is the results. What results are obtained? So NTA, which is nanoparticle tracking analysis. This showed a mean size of 127 nanometers for Eurex with a particle concentration in the range of 10 to the power 8 particles per microliters. So let's see a graph. Here we can see a graph. Here are 4 samples shown. So the first sample, we will be only talking about the first sample. So in the first sample, this is written UC. UC stands for ultracentrifugation. Here we can see that the particle size is 127. This means that through NTA, they have identified that the particle size is 127. What is the particle? The exosomes. The exosomes diameter size is 127 and its concentration is nearly 2. Okay? Not exactly, but nearly 2. In all the other samples, where's my arrow? Okay, I've got it. So in all the other samples, we can see that the diameters are much more higher, but this is the most prominent data which has been retrieved from all the 4 samples. Okay? And this is the most prominent and the most accurate data retrieved. Okay. Next point, bead-spaced flow cytometry confirmed the presence of major component TFR, transferrin receptor, or CD71 on Eurex. This is a biomarker, as I already mentioned in the previous slides. In this graph, as you can see, again, the sample obtained from ultracentrifugation shows the abundance of CD71 most among all the other samples. Here something Alexa 488 is written. This Alexa 488 is basically the fluorescence dye which is being used. So if to explain further, bead-spaced flow cytometry basically uses the phenomenon of scattering of light in fluorescence. So they use fluorescence dyes which proteins absorb, and then it will be easier to identify when light is put on the sample. It will be easier to identify which proteins have absorbed that dye. So here CD71 has efficiently absorbed this dye, and we can see that There is a high abundance of this protein in the ultra centrifugated sample. Okay. It was also confirmed by immunoblot techniques that TFR is present on Hurex. Of course, through the immunoblot techniques, they had confirmed that this protein was also present on the human reticulocyte exosomes. Okay. Now, the third picture which is given here, this is basically the TEM image given. And it was measured here that the diameter of the exosomes calculated from this TEM is 70 nanometers. So one can ask that through NTA we calculated diameter of exosomes, it came 127, and now we are doing TEM, but it is coming 70. Why such a huge deflection? So to answer this question, when TEM technique is used, the sample size reduces. It forms into a pellet for which the diameter also reduces. It is not spread anymore. But in this one, that is for the NTA analysis, the sample is not shrink. The sample doesn't shrink, for which the diameter is a bit expanded. So one can ask which is a more accurate data. So the most accurate data of all here is the TEM image. Okay. Let's move on. So now comes the protein analysis. What proteins are found here in the exosomes? So in Hurex, a total of 367 different proteins are found, including TFR, and 45 unique peptides were also identified. So previously, scientists knew that only there were 7 proteins present in Hurex, but after this research, it was proved that there is 367 proteins present. Next is ATP1A1, ATP1B3. What are these? These are Na plus K plus transporters ATPases. Okay. And SLC1A5 and SLC43A1, these are neutral amino acid genes. These are neutral amino acid genes, which were also identified. So we can say that several Na plus K plus transporters and several neutral amino acid transporters were also identified through the proteomic analysis, and these were present in the exosomes. And these were removed from the reticulocytes when it was maturing into erythrocytes. Glucose 6 dehydrogenase, glycerol 3 phosphate dehydrogenase, these are enzymes, we all know. And SLC6A9, SLC7A1, what are these? These are glucose transporters. These are glucose transporters which were found in the exosomes, okay, which were removed from the reticulocytes when they were maturing into erythrocytes. So a large number of enzymes and large number of transporters were identified through the proteomic analysis of these exosomes. Another very important chemical which was identified here was acetylcholinesterase, which was a notable exception here. In the diagram, that is the pie chart given on the right, we can see that it is written distribution of the proteome of Hurex in subcellular location categories retrieved from UniProt database to gene ontology annotation. So the proteins which were retrieved from the exosomes, it was checked with the UniProt database that these proteins are also present in subcellular location, but in different percentages. Like 25% is present in the extracellular region, 16% in the cytosol, 15% in the plasma membrane, 14% in the nucleus, and like that. Okay. Next, we move on to, let me just move myself away a bit. First, what is since what is visible? Okay, now I think it's visible. It was also identified that dendritic cells can capture Hurex. This is a most prominent finding out of all, that Dendritic cells captures Hurex. Dendritic cells are involved in immune responses, and it was found that these cells capture Hurex. After conducting functional assays, it is seen that Hurex DIL, which is Hurex when treated with chemicals, resulted in Hurex DIL, another sample. Now, this sample was actively taken up by the dendritic cells. Hurex and HIV-1 VLP were in the same cellular component. Hurex uptake was inhibited by monoclonal antibody of singlet 1. That is, you can see this picture right here, right? The black images here, right? So here you can see the red here. The red signifies HIV-1 VLP and the green on the sides is the Hurex. Now this is my arrow. The arrow is keeps getting disappearing. Whatever, I cannot find my arrow on the screen. So sorry. So let me just explain. The green portion is Hurex and HIV-1 VLP is the red part. Now, if this comes in contact with the dendritic cells are not absorbing this, but they are absorbing Hurex DIL. Question is why? Because HIV-1 VLP is an antigen and singlet 1 is an antibody. Of course, the antibody will not let absorption of the antigen. Okay, the monoclonal antibody singlet 1 will not allow the absorption of any component which has the antibody will not allow the antigen to get absorbed, right? So though there is Hurex present, that will not get absorbed. But Hurex in the absence of HIV-1 VLP, that is, you can see Hurex DIL, will definitely get absorbed by the dendritic cells. This clearly proves that Hurex participates in immune responses. Okay, Hurex participates in immune responses and can be further used for various therapeutic applications and can also act as a biomarker in future, alternative biomarker in future for various kinds of diseases. So the key findings, capture of Hurex by dendritic cells indicates a potential pathway for immune communication and regulating the immune system dynamics. Exosomes act as potential biomarkers for early diagnosis, as I already mentioned. So the conclusion which we draw from these are, the report provides the first comprehensive mass spectrometry-based proteomic profile of the human reticulocyte-derived exosomes. It includes identification of proteins in exosome biogenesis. It sheds light that Hurex can present pathogen peptide antigen via dendritic cells. And comparative analysis of the molecular composition of REGs signifies that it can be used as an alternative identifying biomarker. Learnings, we got to learn about exosome biogenesis. We got to know isolation and characterization techniques like UC, ultracentrifugation, and SEC, size, exclusion chromatography. We got to learn about proteomic analysis, immunoblotting techniques, mass spectrometry, immune modulation and possible therapeutic applications, the absorption of Hurex DIL by dendritic cells, confirms that it has answers to the immune responses, right? And it therefore can be used for regulating the immune system dynamics. Gene ontology analysis. The gene ontology analysis introduced us to various proteins and their genes which were identified in the exosomes and which plays a crucial role in the living body. So this is about my research plans. So after I was very much influenced by the finding on absorption of Hurex by dendritic cells and inhibition of absorption of Hurex when it is in composition with HIV due to antibody singlet 1. It fascinated me very much. So I am interested to explore the therapeutic applications of exosomes in various disease models. And it is it has to be tested, its efficacy has to be tested, whether it can be used in autoimmune disorders, for cancers, or any genetic disorders also it can be used or not. Also, I have to decipher in future the evaluation of the delivery methods, the doses in which it has to be given to patients, and also whether it's really working, what are the therapeutic outcomes, how much positive impact it has. So I have such plans in future to broaden this field of study, and I will thank everyone who had given me this chance of presenting such a wonderful review on such a great topic. Thank you so much for this opportunity. Thank you.